Chlamidia. Chlamidia is just a genus in a family, Chlamidiaceae, and the members of this family, they share a common characteristic, specific characteristic as well as unique life cycle. And within Chlamidia as a genus, we find 4 species, and those species are Chlamidia pecorum, Chlamidia pneumoniae, Chlamidia psittaci, Chlamidia trachomatis. Except Chlamidia pecorum, they have associated with human disease. So Chlamidia pecorum is not associated with human diseases. So the family Chlamidiaceae now consists of 2 genera, which is Chlamidia as a genus, which to include Chlamidia trachomatis, and Chlamydophila to include Chlamidia pneumoniae, and Chlamidia psittaci, and Chlamidia pecorum. But Rickettsia is just a genus, and the Rickettsia is a genus in order Rickettsiales, which they include two families. In Rickettsia, there are 2 families, and the first family is called Rickettsia, and the second family is called Anaplasmataceae. So these are the two families in Rickettsia. So the family Rickettsia then includes Rickettsia and Orientia, whereas family Anaplasmataceae include Rickettsia, Anaplasma, Cohudria, Rickettsia, as well as Ozatia. So these are genera in family Anaplasmataceae. So Chlamidiaceae is, of course, we have Chlamidia as genera. Chlamydia are deficient in energy metabolism, and therefore they are obligate intracellular parasites. So they are not able to metabolize their own energy source. So that's why they are obligate intracellular parasites. And the unique growth cycle involves 2 distinct forms. We have elementary body of Chlamidia, as well as reticulate body, and the elementary body is infectious, whereas reticulate body is not infectious. So these are the 2 distinct forms in life cycle of Chlamidia. So elementary body has outer membrane similar to that of many gram-negative bacteria, and the Chlamydia outer membrane also contains lipopolysaccharides, which is called the LPS, as well in the membrane of these or outer membrane of these Chlamydia. So the first Chlamydia that is recognized as causative agents of human infections is Chlamidia trachomatis, and the Chlamidia trachomatis, of course, has also been divided into 3 different biovariants. And talking about biovar means that the organism has got 3 biological variants, or 3 main biological variants, which is trachoma, the one that causes trachoma, and the one that causes lymphogranularoma venereum, and also the other which causes mouse pneumonitis. So these are the 3 variants or bivariants of Chlamidia trachomatis. I don't know if Sevia has a question, because I can see her raised hand. If you have a question, kindly you can ask your question before we proceed with the Chlamidia trachomatis. Okay. Okay, that's all right. Thank you. So now we have 3 biovariants. So biovariants, as you remember, we're talking about, we have what you call cellovars or biovars. So biovariants means these are the 3 biological organisms or biological life that are associated with different types of life or different types of diseases. For example, we have one biovariant of Chlamydia which causes trachoma. This affects eyes, and the second variant is called lymphogranularoma venereum, which of course affects respiratory reproductive organs, and the mouse pneumonitis that affects respiratory tract, which is of course more specifically lungs. So the trachoma biovar include also cellovars, and talking about cellovar means means serological variants, which means like different serological markers, like different antigens, and just call them antigens among one biovirus. So biovirus can be a very broad group of organisms associated with the one type of disease, but serovirus means among them there will be some differential characteristics according to their serological markers, like antigens. So we have serovirus from A through K of trachoma, and the serovirus A, B, and C are associated with the severe eye infection, which is called trachoma, but serovariant D through K, I, and J are associated with inclusion conjunctivitis and urogenital infection, whereas serovariant L, L1, L2, L3 associated with lymphogranuloma venereum, which is called LVG, and DV, and invasive urogenital tract disease. Of course, this is what we call lymphogranuloma venereum. So clinical infections are associated with the Chlamidia trachomatis. You find the first one is called trachoma, and trachoma is of course infection of eye. This is chronic eye infection. The number one cause of preventable blindness in the world. So this is the first or number one cause of blindness which can be prevented. So it is called trachoma, and trachoma is associated with the serotypes A, B, and C. These are the serovariants A, B, and C associated with trachoma. So these serovirus produce a chronic infection resulting in scarring of the eye and continual abrasion of the cornea of the eyelid turns downwards towards the cornea, and left untreated, infection can end up causing blindness in adults. So just because of the scarring of the cornea and the abrasion of the cornea as the eyelid turns downwards toward the cornea, so it can cause serious blindness like this. You can see the scarring, the conjunctival scarring of conjunctiva, and of course this can lead to blindness. So that is what you call trachoma of the eye. So the second infection caused by Chlamidia trachomatis or infectious disease is called Chlamidia. It's called lymphogranuloma venereum. Of course, it's caused by Chlamidia trachomatis serovariant L to L3. This is what you call lymphogranuloma venereum. It is a sexually transmitted disease, and the serovirus are more invasive than others. So the lymphogranuloma venereum patients, they have inguinal and anorectal symptoms, and of course these serovirus are able to survive inside the mononuclear cells, and you find that bacteria would enter lymph nodes near their genital tract and produce strong inflammatory response that often results in buboes formation and subsequent rupture of the lymph nodes. So you find that there would be buboes, the buboes or buboes formation, of course, around the inguinal lymph nodes near the reproductive system organs. Like this, you can see this is the inguinal swelling of the lymph nodes or the lymphatic drainage. In inguinal lymphatic drainage, you see the swelling. So that will be it's called buboes for the LGV, which is lymphogranuloma venereum serovarians L of Chlamidia trachomatis. So Chlamidia trachomatis can also cause other urogenital disease. For example, Chlamidia trachomatis can also be associated in men. For example, in men is associated with non-gonococcal urethritis in men. And you remember, this was my question last time in assignments. I was asking, for example, if you are able to see many mononuclear MPN or polymorphonuclear cells in the swabs, swab or vaginal swab, and especially in men, but without any gonorrhea, what would you expect? You'd expect non-gonococcal urethritis, and this is actually associated with Chlamidia trachomatis. You find also it can cause epididymitis of epididymis, as well as prostatitis of the prostate organ. And those serovariants are just through D through K, D, F, D, E, F, and K. So the same serovirus can also cause conjunctivitis in male and female. So infections in adult women, apart from men, can cause urethritis, follicular cystitis, endometritis, bronchitis, laryngitis, pelvic inflammatory disease, as well as perihepatitis. So these are the infections in women. And cystitis, of course, can lead to scarring and dysfunction of the oviductal transport system, which can result in ectopic pregnancy. Infertility and ectopic pregnancy are associated with the dysfunction of the fallopian tube, which is called oviduct. So another condition associated with the Chlamidia trachomatis, it is Reiter syndrome, which is associated with inflammation of the urethra, which is urethritis, conjunctivitis, polyarthritis, as well as other regions of the mucocutaneous. And apart from Reiter syndrome, we have also Chlamidia infection in the newborn. Newborn will also suffer Chlamidia infections, especially upon traveling through infected birth canal. An infant can be infected with the Chlamidia species, especially when they are traveling through infected birth canal of the mother, pregnant women. And infants suffering from Chlamidia infection can experience serious manifestations, including conjunctivitis, nasopharyngeal infections, as well as pneumonia. So the incubation period is a variable, but symptoms will generally appear within 3 weeks after birth. That's where you find that children are actually exhibiting symptoms like conjunctivitis symptoms, as well as pneumonia and also nasopharyngeal symptoms. So lab diagnosis for Chlamidia, of course, include that specimen it depends on symptoms, because you have seen that Chlamidia trachomatis is associated with various syndromes, various infections. Infection of the eye, of the genital tract, genital organs, as well as pneumonia, of course, and many other infections are associated with it, of course, eye and so on. So you find that symptom like specimen depends on the symptoms that are presenting. So the specimen should consist infected epithelial cells and not egg exudates. So don't collect egg exudate alone. Make sure that you collect epithelial cells, because this organism is intracellular, can survive intracellularly in the epithelial cells and also, of course, white cells. So first void urine and vaginal swab are excellent for detecting of the infection. So first void urine, that is morning specimen. So first, after receiving specimen in lab, then you can proceed with the direct microscopic examination, where you, of course, you perform cytological methods. Of course, that is direct cytological examination, which involves trachoma and inclusion conjunctivitis, and is sensitive in detecting ocular infection in the newborn, especially observing for a trachoma and inclusion conjunctivitis. So those are some of the inclusion bodies you find within the cells. This is intracellular inclusion body, especially taken from ocular infection or ocular swab of a newborn. You find that this is the inclusion bodies of Chlamidia. So this confirm when you perform eye culture, it can confirm Chlamidia trachomatis. Otherwise, you find the inclusions within cells, intracellular inclusions. So apart from that, also we can perform direct fluorescence antibody technique, which is called DFA. It is used for endocervical or urethral specimen, and direct examination of cytological stain of endocervical smear can show also inclusion bodies in columnar epithelial cells. You find very large inclusion bodies of Chlamidia trachomatis. Of course, using a pap stain, pap smear, pap stain, you can demonstrate inclusion bodies within these endocervical epithelial cells and of course, columnar epithelial cells like this, and see the inclusion bodies within columnar epithelial cells. infected. So because it is not easy to grow chlamydia outside of the cell, it is not possible at all to grow chlamydia outside of the cell. Then it is very important to perform cell culture. Of course, you can perform cell culture, of course, including McCoy, human epithelial cell, 2 HeLa cells, buffalo green monkey kidney. You can use either of these to grow chlamydia species. So the cell lines are grown on cover slip in 1 drum shell vial on the surface of the Matwell cell culture dish that contains cell culture media with chlorhexidine. So then the specimen is then cytocentrifuged onto the cell monolayer and incubated for 72 hours. And fluorescence labeled monoclonal antibodies can be used to detect inclusions. So we can use, of course, fluorescence labeled monoclonal antibodies because we want to use fluorescence microscopy in order to observe those inclusion bodies. Otherwise, iodine or GM sustain can be used to observe intracellular inclusion bodies of chlamydia. So they can be shown like this, as you can see, intracellular inclusion bodies within this the one which is iodine stained, and also the infected McCoy cells that we use for culture. You can see the intracellular inclusion of the Chlamidia trachomatis, and you can see the iodine staining. You can see the inclusion bodies within the infected cells, the McCoy cells. So the half moon of the half moon shape of the inclusion can be seen within this cell for culture. So immunoassays can also be used to detect antigen, and antigen to be detected by enzyme immunoassays is outer membrane lipopolysaccharides, which is called LPS. Outer membrane LPS chlamydia antigen can be detected, or we can detect major outer membrane protein, which is called MOMP. So these are the 2 antigens that can be detected on Chlamidia trachomatis using immunoassays, especially enzyme immunoassay. So detect outer membrane lipopolysaccharides or the major outer membrane protein, or MOMP. So chlamydia pneumoniae is another chlamydia that is associated more specifically with acute respiratory diseases. It's not just about other sexual transmitted disease. These are only acute respiratory disease and pneumonia, and also pharyngitis. So this chlamydophila pneumonia, it affects only the respiratory tract. It also has been isolated from patients with otitis media. So otitis media, of course, with the effusion, pneumonia with the pleural effusion, as well as aseptic pharyngitis. So otitis media is, of course, one of the conditions, especially in young children. They are normally affected, they are highly affected with the otitis media, especially with other conditions apart from this. Also, we have also seen Haemophilus influenzae can also cause otitis media. So, but also, of course, otitis media, chlamydophila pneumonia is also highly and common. It is also a risk factor for Guillain-Barré syndrome, asthma, and cardiovascular disease as well. So, infections associated with the chlamydophila pneumonia include, of course, that will result in a prolonged sore throat and hoarseness, and followed by flu-like lower respiratory tract symptoms. And then second phase of illness is biphasic, which results in pneumonia and bronchitis, and sometimes sinusitis. So these are some of the infections. Of course, all the infections associated with the chlamydophila pneumonia are all respiratory tract infections, from the lungs all the way through the pharynx and nose. Of course, sinusitis. Lab diagnosis for chlamydophila pneumonia, we can collect either specimen, but specimen for chlamydophila pneumonia are just specific for respiratory tract: sputum, bronchial washings, or bronchial lavage fluids, nasopharyngeal aspirate, throat washings, and throat swabs. They are all associated. They can all be used for isolation of chlamydophila pneumonia, and the chlamydophila pneumonia can also be cultured on cell lines, and they will be also visualized using fluorescent conjugate monoclonal antibodies. This is also the same thing as well. So it is not like you are going to use just normal culture media to isolate them. We use cell lines and we have to visualize them the same way using fluorescent conjugate monoclonal antibodies. So you can use these cells, human epithelial cells, or monoclonal antibodies can be used to detect them as inclusion bodies in human epithelia cells, the HEP2 and human lines. Using, of course, direct, indirect fluorescence antibody to detect antibodies against these pneumonia in respiratory secretions, and the antibodies will, of course, detect the OMP, that is the protein that will be detected, outer membrane proteins. So that is the protein that will be detected, and because of the fluorescence, you can see that the inclusion bodies within cell lines or cytoplasmic inclusion bodies that you find within cell lines we use for culture, they can fluoresce, and when you are using fluorescence microscope, you could be able to see fluorescing inclusion bodies within human epithelial cells, of course, and also many other cell lines you can use to isolate chlamydophila pneumonia. So that's about chlamydia. So you can see that chlamydia is actually very, very, very strange organism, very strange. So you can sometimes, it is very hard to isolate, it's very hard to grow, it's very hard to treat in lab. So because they are very specific bacteria, so they don't exhibit many other characteristics of other bacteria. But because they are almost the same as Rickettsia, that's why we tried to group them together in one session, in one presentation, so that when you are reading, you'll be able to have them together all. So Rickettsia and many other organisms. So we find Rickettsia as a genus, and Orientia, they are all belong in the same family, which is Rickettsia. And most member, they are arthropod-borne and obligate intracellular pathogen, which can only grow in the host cells. So, which means also Rickettsia is going to be the same as Chlamydia, because as you can see, we have made a very good arrangement in order to make sure that we arrange them together in the same document, because they both exhibit almost the same characteristic, being intracellularly and obligate intracellular organisms. So Rickettsia are just short and non-motile gram-negative bacilli, and Rickettsia has never been grown in cell-free media, but have been grown in yolk sac of embryonated eggs and monolia cell lines. So Rickettsia, we have to divide the Rickettsia into 3 groups depending on the condition they are associated with. Typhus group contains 2 species, Rickettsia prowazekii and Rickettsia typhi, as well as spotted fever group that contains Rickettsia rickettsii, Rickettsia connerii, and Rickettsia africae. Because infective aerosol dose is low, Rickettsia rickettsii, Rickettsia prowazekii, Rickettsia typhi, and Rickettsia connerii, all of them are considered potential bioterror agents. So these are agents of bioterrorism. So Rickettsia rickettsii, Rickettsia prowazekii, Rickettsia typhi, and Rickettsia connerii. So because, of course, their infection dose is very low, any small amount of infection in the aerosol can be very effective in the Rickettsia. So the first condition that we normally know in Rickettsia or associated with Rickettsia is a spotted fever group or disease, where you find Rickettsia, Rickettsia connerii, and africae. This is a spotted fever group which is called Rocky Mountain spotted fever. Rocky Mountain spotted fever is the most severe form of Rickettsia infection. and Rocky Mountain spotted fever is most commonly associated with Rickettsia rickettsii, and it is just a zoonosis disease, of course, because these infections are transmitted by tick bites from other animals, and tick bites, of course, for example, Chlamidia variabilis and Chlamidia andersoni, they are all tick that can transmit these Rickettsia rickettsii through tick bites. So this is a dorsal view of Chlamidia variabilis, which is a vector for Rocky Mountain spotted fever transmitted by Chlamidia andersoni and Chlamidia variabilis through bites. Once the host tissue, in the host tissue, the Rickettsia are just phagocytosed and into endothelial cells, and they can just replicate in the cytoplasm of endothelial cells, right? Of course, endothelial cells are the cells of the vessels, blood vessels, and in the host cells, they can pass directly through plasma membrane into adjacent cell without causing damage, and the patient experience flu-like symptoms: fever, headache, myalgia, and nausea, and vomiting and rash. So once disseminated, of course, the organism can cause vasculitis. So vasculitis is inflammation of the blood vessels. So those vessels affected are vessels in the lungs, brain, and the heart. So pneumonitis is also very common, and CNS manifestations and myocarditis can also occur, which leads to hypovolemia and hypotension and DIC. So these are the symptoms of Rocky Mountain spotted fever. You find that someone begins with the flu-like symptoms, but will extend, even causing problems in the lungs and the brain because of vasculitis in those organs. The second disease associated with the Rickettsia is Boutonniere fever. Boutonniere fever, it is also known as Mediterranean spotted fever. It is caused by Rickettsia conurei, and this condition is very common in France and Spain and Italy. And the Rickettsia conurei has also been causing disease, has also been causing what we call Kenya tick typhus, and South African tick fever, and Indian tick typhus. So these are some of the typhus conditions that have been found in these countries: Kenya, South Africa, as well as India. So, but of course, this organism is very common in France, Spain, and Italy. So the Boutonniere fever is normally similar to the Rocky Mountain spotted fever, but the difference is that rash involves palm of the hands and soles of the feet, just as in Rocky Mountain, but the rash involves the face as well. So, but face is not involved for Rocky Mountain spotted fever. So, in contrast with Rocky Mountain, the disease is characterized by the presence of tache noire. So, tache noire is what you call black spots as the primary site of infection. So, if the primary site of infection is maybe face or trunk, you find tache noire. So tache noire are just French name. It's just like translation from French to English. That is tache noire, which means black spots. So you find the black spots at the site of infection. So that is maybe trunk, depending on the tick bites, where the tick bite took place. So that's where you find also those tache noire or black spots there. So typhus group of organisms you find apart from, of course, spotted fever or Boutonniere fever, you find also typhus group. You will be able to see species like Rickettsia typhi that cause murine typhus, Rickettsia prowazekii which cause loose bone typhus, and Breyer's Zancel disease. And the typhus Rickettsia differ from the other Rickettsia group in the replicate in the cytoplasm of the whole cell called cell lysis, thereby releasing Rickettsias. Other types of Rickettsia, they don't cause cell lysis. They can just exit cell without causing lysis. But typhus group, they can rise cell before they exit from one cell to another cell. So one of the conditions is called murine typhus, and this is actually transmitted by a rat flea, which is called Xenophila cheopis, and human can be infected only when fleas defecate on the surface of the skin while feeding. And if someone is going to scratch the site, if you are scratching the site, then you can allow direct inoculation into the abrasions on your skin. So that is because of scratching. When you are feeling like sometimes like pain because of that feeding of the fleas, you can feel that kind of scratch. And when you're scratching, that kind of pain, when you scratch it, then you're going to allow inoculation of these feces into abrasions. So that's how you can get then Rickettsia typhi in your body. So Rickettsia typhi can also be transmitted by human directly into from the flea bite itself, without needing of depositing maybe feces, and you need to scratch the site. Even if fleas can bite and introduce infection immediately into your body, as it is the case of Rocky Mountain. Clinical cause of endemic typhus include fever, headache, and rash. But unlike Rocky Mountain, you can differentiate that rash production is relatively rare, and when present, they usually occur on the trunk and extremities. But rash on the palm of the hands also can occur. So, but Lowe's-borne typhus or epidemic typhus is caused by Rickettsia prowazekii, and the vectors include human louse, which is called Pediculus humanus, and skilled field, which is called Okepias huali, and skilled louse, which is called Neohematopinus scuropelli. So these are the fleas that are on, or the louse, the lice and fleas that are associated with these louse-borne typhus. So transmission is that the same as murine typhus, because murine typhus is just associated with Rickettsia typhi, because those are the thick bite, the fleas bite, not loose, because these are the loose, whereas Rickettsia typhi is just transmitted by fleas. So the disease is just similar to that of Rocky Mountain, but you find that involvement of palm of the hands and soles of the feet with rash. But unlike some cases of Rocky Mountain, the face may also be affected by rash. But remember that face is not affected in Rocky Mountain spotted fever. So general lab diagnosis for Rickettsia disease normally, we have seen sometimes, of course, because of their infection nature, isolation of Rickettsia is not recommended. So it is not recommended to isolate Rickettsia. You can only, when you want to isolate them, you can only attempt this isolation at least in biosafety cabinet level 3 laboratories. You know, these level 3 laboratories, we don't have them many places. In most of our hospitals, we have level 2. If you remember what we normally describe in accreditation, for you guys who have been dealing with accreditation, you know that level 3 is very advanced. So level 3 is used only for very highly infectious diseases. Where, like for example, TB, we can use level 2 for TB. TB is fine to use level 2, but level 3 can be used for very high infectious diseases. If culture is then attempted, if you want to attempt a culture, then blood should be collected as early in the disease as possible, and immunohistochemistry detection of Rickettsia can be used, but typically serological assays are the only laboratory tests performed for diagnosis of Rickettsia. So we don't need to isolate them in lab, right? So the immunofluorescence antibody technique is the most considered technique and is a gold standard to detect antibodies to the Rickettsia, because there's no need of isolating them in lab, because even handling them is not easy. And the reason is that you know that Rickettsia prowazekii, Rickettsia typhi, Rickettsia connori, all of them, they are bioterrorism agents, because there's only a very small need, a very low need of dose, or a very low dose that can establish disease. So that's why they are really very, very serious infectious pathogens. So we should try our best. If you want to isolate them, use level 3 laboratory if necessary, but mostly we don't need that. We only need to isolate them or to test them using immunofluorescence antibody testings. So that's how it is for both Rickettsia species and the Chlamidia species and similar organisms. I know you can see that their common feature and their unique and common feature between the two genera. We find that they are intracellular organisms, and they are not able to synthesize or to metabolize their own energy for use. So they need to be grown, of course, in cell lines if they are grown. But of course, some of them are even bacterial agents. It is recommended to be very careful when you are trying to isolate them. So we have other bioterrorism agents. You remember, we have many other. We discussed them. We have Pastorella, we have Francisella, we have so many. You know what we call Tularemia or Francisella tularensis. That is also one of the bioterror agents. Even Pastorella, even Brucella, all of them are bioterror agents. So I think we have that class. I think it was yesterday. No, a day before yesterday, because yesterday we had anaerobes. But before anaerobes, remember, we had also like Pastorella, Francisella, Brucella. We have all of them. So all of them, they are elements of bioterrorism. Even Rickettsia. So if you go through notes, you'll see them. They are there. So thank you so much for your time. God bless you, and see you tomorrow. So we are trying to upload these videos that we are recording, and we'll be just recording slowly. So, but it is also very important now to attend and we'll have discussion together. So thank you. God bless you, and see you tomorrow for the next presentation.